In this video, we discuss how both speed and sample loading volume and size exclusion chromatography can be increased using a novel chromatography device developed by researchers at McMaster University. Size exclusion chromatography, or SEC, is widely used for the analysis of biological macromolecules. New studies have shown that SEC could potentially be used for the preparative purification of biologics such as mRNA, DNA, viruses, virus-like particles, and vesicles that can be isolated in the void fraction of appropriate SEC columns. Unlike techniques such as ion exchange, affinity, and hydrophobic interaction chromatography, SEC does not involve change in buffer composition during separation and is therefore thought to be better at ensuring stability of complex biologics. However, unless some of the limitations of conventional size exclusion chromatography columns, such as low speed, low loading capacity, and poor scalability, are addressed, SEC will have relatively limited use in large-scale purification of these new generation biologics. At McMaster University, we have developed novel cuboid packed bed chromatography devices to address problems such as speed, sample loading capacity, and scalability in process chromatography. These box-shaped chromatography devices that have relatively short bed heights and large cross-sectional areas have already been shown to be suitable for fast and scalable, high-resolution, ion exchange chromatography, and size exclusion chromatography of proteins. For technical details, please read the papers listed here. First, we have a short video clip showing a cuboid SEC device. This prototype device was fabricated by 3D printing. The resin was packed by a simple flow packing technique. The inlet and the outlet of this device were located spaced diagonally with respect to each other. The liquid was fed to the resin bed and collected from it using complementary sets of primary and secondary channels located at the top and at the bottom of the device. These flow distribution and collection features, which are discussed in detail in the publications listed in the previous slide, ensure that the flow of liquid in the cuboid SEC device is uniform. Flow uniformity is critically important in chromatography. The cuboid SEC device was integrated with a Bio-Rad-NGC liquid chromatography system. Next, we present an experiment to demonstrate fast preparative separation of a binary model protein mixture using our prototype cuboid SEC device at a very high load condition. This video has been sped up 40 times in order to keep the overall length of this presentation short. The 200 ml device was packed with cefacryl S200 resin. The flow rate used in the experiment was 20 ml per minute, which corresponded to 0.1 column volume per minute. The volume of protein sample loaded was 40 ml, which corresponded to 0.2 column volume. As can be seen in this video clip, excellent separation of the two model proteins could be obtained using our prototype 200 ml cuboid SEC device even at very high overload and very fast flow conditions. The separation was complete in less than 30 minutes, which is quite fast for preparative SEC of proteins. Conventional SEC columns are typically operated at less than 0.01 column volume per minute. Our cuboid SEC devices can easily be operated at 0.1 column volume per minute. Therefore, the flow rate and the productivity could be increased by a factor of 10. The typical sample loading volume in a conventional SEC column is 0.04 column volume. With our cuboid SEC devices, the sample loading volume could be as high as 0.2 column volume. Therefore, the sample loading capacity could be increased by a factor of 5. We have also used our cuboid SEC devices for purifying very large proteins like antibody aggregates, viruses, and virus-like particles, and look forward to sharing these results with you once these are published. Thank you for watching. We look forward to your feedback and comments.